For most of the history of biology, scientists worked from a set of quiet, largely unstated assumptions about what life actually requires to exist, assumptions so deeply embedded in how researchers thought about biology that they rarely needed to be stated outright at all. Life needed sunlight, or at least needed to exist within a food chain that traced back to sunlight somewhere. Life needed liquid water, in reasonably moderate temperature ranges, not boiling and not frozen solid. Life needed protection from radiation, needed a reasonably stable chemical environment, and needed some meaningful supply of organic nutrients to sustain itself. These assumptions weren't reckless or unscientific. They were built on centuries of careful observation of the plants, animals, and microorganisms that dominate the visible surface of the planet, in the places humans have always lived and studied most closely. The trouble is that every single one of these assumptions has now been broken repeatedly by discoveries made across wildly different environments over the past several decades, to the point where the phrase life as we know it, once used as a reasonably confident boundary marker for what biology could and couldn't do, has become something closer to a standing joke among researchers who study extreme environments, a phrase invoked mainly to be immediately followed by an example of life doing exactly the thing it supposedly couldn't. The clearest starting point for this story is the discovery of hydrothermal vent ecosystems in 1977, an event that arguably did more than any single other discovery to permanently dismantle the assumption that all life depends, directly or indirectly, on sunlight. Researchers aboard the submersible Alvin, exploring the Galapagos Rift along the ocean floor, expected to find a barren volcanic landscape, since nobody had any serious reason to expect abundant life clustered around superheated mineral vents in complete darkness, at depths exceeding 2,000 meters under crushing pressure, in water that could exceed 300 degrees Celsius near the vent openings themselves. Instead, they found dense clusters of life unlike anything cataloged before. Enormous tube worms lacking mouths or digestive systems entirely, eyeless shrimp swarming in the hundreds, ghostly white. Crabs and mussels, packed together in numbers that would have been impressive even in a nutrient-rich shallow reef, let alone in what should have been one of the most hostile, barren environments on the planet. The explanation that emerged from subsequent research reshaped fundamental biology textbooks. These ecosystems run on chemosynthesis rather than photosynthesis, with specialized bacteria converting chemicals like hydrogen sulfide, extremely toxic to most surface life, into usable biological energy, and larger organisms either hosting these bacteria directly within their own tissue or feeding on bacterial mats that coat the surrounding rock. An entire complex food web, supporting large, visually striking animals, existing independent of the sun in total darkness, was not supposed to be possible according to the biological consensus that existed the day before this discovery. And the fact that it clearly was possible forced an immediate and permanent revision to how scientists thought about the fundamental requirements for complex life. If hydrothermal vents demonstrated that life didn't need sunlight, research into the deep subsurface biosphere over the following decades demonstrated something arguably even stranger, that life doesn't need to exist anywhere near the surface at all and can persist for extraordinarily long periods, sealed away in solid rock kilometers beneath the ground, in conditions that would seem, on first inspection, to leave no room for anything resembling a habitable environment. Researchers drilling boreholes for mining exploration, groundwater studies, and dedicated microbiology research have repeatedly found living microorganisms in rock formations several kilometers underground, far below any plausible connection to surface ecosystems, sunlight, or recently deposited organic material. One of the more remarkable examples comes from the Kidd Creek Mine in Ontario, Canada, where researchers studying water trapped within ancient rock formations roughly 2.5 kilometers underground found water that had been isolated from the surface for an estimated 1 to 2.5 billion years, based on the ratios of noble gas isotopes present in the water, which serve as a kind of geological clock for how long a water sample has been sealed away from surface exchange. Remarkably, this ancient, isolated water contained clear evidence of microbial life, apparently surviving on chemical energy derived from reactions between the water and surrounding rock, specifically a process involving radiolysis, where natural radioactivity in the rock splits water molecules and generates hydrogen and sulfate compounds that microorganisms can use as an energy source without any input whatsoever. From sunlight, photosynthesis, or surface-derived organic matter across a time span that predates the emergence of complex multicellular life on the surface of the planet by well over 1 billion years. The scale of this deep subsurface biosphere, once researchers started taking it seriously enough to actually estimate its size, turned out to be almost unbelievably large. A major international research collaboration called the Deep Carbon Observatory, which spent roughly a decade coordinating research across disciplines to better understand the scope of life existing deep within Earth's crust. published estimates suggesting that the total mass of microbial life living within Earth's deep subsurface, encompassing both continental rock formations and marine sediment beneath the ocean floor, could rival or even exceed the total biomass of all life living on the surface of the planet combined, including every plant, animal, and surface microorganism that exists in what most people would consider the normal, visible biosphere. This is a genuinely difficult claim to fully absorb because it suggests that a comparable or greater amount of total living material, in terms of raw mass, exists hidden within solid rock and sediment in conditions of total darkness, often at extreme pressure, sometimes at elevated temperature, surviving on chemical energy sources rather than sunlight, compared to everything visible and familiar living across every forest, ocean surface, grassland, and city on the planet. If this estimate holds up as more research refines it, it means that for the entire history of biology, as a scientific discipline, researchers have been building their understanding of life on Earth almost entirely by studying what amounts to a minority, rather than a majority, of the planet's total living material, simply because the majority happens to live somewhere humans historically had no way of directly observing. Radiation resistance provides another category of discovery that has repeatedly forced revisions to what scientists assumed life could tolerate. And the poster organism for this particular category is a bacterium called Deinococcus radiodurans, first identified in the 1950s, after researchers found it surviving in canned meat that had been irradiated with doses of radiation. Intended specifically to sterilize the food completely by killing every living organism present, Deinococcus radiodurans turned out to be capable of surviving radiation doses thousands of times higher than what would be lethal to a human, and considerably higher than what would destroy the DNA of essentially any other known organism beyond repair. Subsequent research into how this bacterium achieves this resistance revealed an extraordinarily effective DNA repair system capable of reconstructing its own genome even after radiation exposure has shattered the DNA into hundreds of separate broken fragments, essentially reassembling its own genetic blueprint from pieces in a way that no other well-studied organism can replicate with anywhere near the same efficiency. This discovery mattered beyond simple curiosity about a resilient bacterium, because it directly informed research into radiation-resistant crop development, waste, cleanup at radioactive contamination sites, and more speculatively, discussions about what kind of life might be capable of surviving. The intense radiation environments found in space or on the surface of planets and moons, lacking the kind of protective magnetic field and atmosphere that shields life on Earth from the worst of solar and cosmic radiation. The Atacama Desert in Chile offers a different but equally striking example of life persisting in conditions long assumed to be fundamentally incompatible with biological activity, this time centered on extreme aridity rather than radiation, pressure, or darkness. Certain regions within the Atacama receive essentially no measurable rainfall for years or even decades at a time, making it, by most standard measures, the driest nonpolar desert on the planet, a place so consistently dry that some researchers have used it as a practical stand-in environment for studying the kind of conditions found on the surface of Mars, since the two environments share meaningful similarities in aridity, soil chemistry, and radiation exposure. For a long time, portions of the Atacama's driest core were considered essentially sterile, too dry to support any meaningful microbial activity at all, a genuinely dead landscape rather than simply an inhospitable one. More recent research, using increasingly sensitive detection methods capable of identifying extremely sparse and slow-growing microbial populations, has found that even the driest parts of the Atacama support microbial communities, organisms that appear to survive by extracting the minimal moisture available from occasional fog from water, absorbed directly into salt deposits, or from thin films of moisture that form briefly under specific atmospheric conditions, too subtle for earlier detection methods to notice or measure reliably. These microorganisms don't merely survive passively in a dormant state during dry periods, waiting for rare rainfall, but in some cases show evidence of remaining metabolically active even during the driest stretches, running their biological processes at an extremely slow pace using whatever minimal moisture theoretically shouldn't be sufficient to support any activity at all, according to earlier models of what minimum moisture threshold living cells require to function. Antarctica's McMurdo Dry Valleys provide a related example, since this region represents one of the coldest, driest, and windiest environments on the planet's surface, so extreme that these valleys are largely ice-free despite their Antarctic location, because the same harsh winds that make the region so hostile also prevent snow and ice from accumulating in the first place, leaving exposed rock and soil that more closely resembles conditions found on Mars than anywhere else readily accessible on Earth's surface. Researchers studying the dry valleys have found microbial life surviving inside. Rocks themselves, in a category of organism called endoliths, which colonize the tiny air spaces and mineral structures found just beneath a rock's outer surface, using the rock itself as a kind of protective shield against the valley's extreme cold, intense ultraviolet radiation, and desiccating winds, while still receiving enough filtered sunlight through the translucent outer layer of certain rock types to support a slow, minimal form of photosynthesis. These endolithic communities grow at an almost imperceptibly slow pace, potentially taking centuries to develop the thin, visible colored bands that researchers can identify just beneath a rock's surface when they crack a sample open, representing one of the slowest growing, most resource limited ecosystems ever documented, and yet persisting reliably across one of the most hostile terrestrial environments available for study, a genuine ecological niche rather than simply scattered, temporary survivors clinging to existence. Hypersaline environments, meaning bodies of water, with salt concentrations far exceeding that of normal seawater, sometimes approaching full saturation, where salt crystals actively form within the water itself, represent yet another category where life persists well past the point where most biology would predict cellular function should become impossible. The Dead Sea, various hypersaline lakes in locations ranging from Australia to Antarctica's own McMurdo Dry Valleys, and artificial salt evaporation ponds used in commercial salt production, have all revealed thriving microbial communities specifically adapted to these extreme salinity conditions. Organisms called halophiles that have evolved specialized cellular machinery to prevent the kind of catastrophic water loss that would normally occur when a cell is placed in an environment with such an extreme concentration gradient between its internal chemistry and the surrounding water. Some of these hypersaline environments take on a visibly striking pink or red color caused by the pigments these salt-loving microorganisms produce as part of their adaptation to the intense sunlight and osmotic stress they experience, turning what would otherwise look like a completely barren, chemically hostile body of water into a visibly, sometimes strikingly colored habitat teeming with a specialized form of microbial life invisible to the naked eye beyond the color it collectively produces. Perhaps no organism has captured public fascination around biological extremity quite as thoroughly as the tardigrade, a microscopic invertebrate, sometimes called a water bear or moss piglet due to its appearance under magnification, that has become something of a mascot for extremophile research, despite technically living in relatively mild environments most of the time, generally preferring damp moss, lichen, and similar moist habitats rather than the extreme conditions it has become famous for surviving. What makes tardigrades scientifically remarkable is not where they normally live, but what they can survive when their normal environment dries out or otherwise becomes hostile, triggering a biological state called cryptobiosis, in which the tardigrade essentially shuts down its metabolism almost entirely, expelling most of its internal water and replacing it with a specialized sugar compound that helps stabilize its cellular structures during this dormant state. In this cryptobiotic condition, tardigrades have been documented surviving temperatures approaching absolute zero, temperatures exceeding the boiling point of water, pressure roughly 6 times greater than found at the deepest point of any ocean trench, direct exposure to the vacuum of space, and radiation doses that would be lethal to nearly every other known animal by an enormous margin. In 2007, researchers actually sent tardigrades into orbit aboard the European Space Agency's Photon M3 mission, exposing them directly to the vacuum of space and unfiltered solar radiation outside any spacecraft shielding, and found that a meaningful percentage of the tardigrades survived this exposure and successfully revived once returned to normal conditions, a result that remains one of the most striking demonstrations available that complex multicellular animals, not just single-celled bacteria, can survive direct exposure to conditions long assumed to be instantly and completely lethal to any form of life more complicated than the hardiest bacterial spore. The tardigrade story took an unexpected and widely discussed turn in 2019 when the Israeli lunar lander Bereshit crashed on the moon's surface during its final descent, carrying, among other cargo, a data archive that included a sample of dehydrated tardigrades in cryptobiotic state, included as part of an initiative intended to preserve a record of human civilization and terrestrial biology in case of catastrophic events on Earth. Following the crash, considerable public speculation emerged about whether these tardigrades might have survived the impact and could now exist on the lunar surface in some dormant state, potentially representing the first instance of terrestrial life reaching another celestial body outside of deliberately controlled experiments. Scientific assessment of this scenario generally concluded that survival, while not entirely impossible given tardigrades' documented resilience, was unlikely given the specific combination of impact forces involved in the crash and the complete absence of any plausible pathway for these organisms to ever revive, since reviving from cryptobiosis requires rehydration, a condition essentially impossible to achieve naturally on the moon's completely dry, airless surface. Regardless of the ultimate fate of that specific tardigrade sample, the episode illustrates how thoroughly public understanding of biological limits has shifted to the point where a crashed spacecraft carrying dehydrated microscopic animals could generate serious, if ultimately inconclusive, scientific discussion about whether Earth life might now technically exist on the Moon. Beyond tardigrades, research into radiation-tolerant life took on a particularly sobering real-world dimension following the 1986 Chernobyl nuclear disaster, when the surrounding exclusion zone became, almost overnight, one of the most intensively studied radioactive environments on the planet, offering researchers an unplanned but scientifically valuable opportunity to study how various organisms respond to sustained elevated radiation exposure over an extended period, rather than the brief, acute exposure typically studied in laboratory settings. Among the more striking findings to emerge from Chernobyl research involve certain species of fungi found growing directly on the walls inside the damaged reactor building itself, an environment with radiation levels high enough to be immediately lethal to unprotected humans within a short period of exposure. Researchers studying these fungi found that some species appeared to actually grow toward, rather than away from, sources of radiation, and further investigation suggested these organisms contain high concentrations of melanin, the same pigment responsible for skin coloration in humans, which in this context appears to help the fungi convert radiation into usable chemical energy through a process some researchers have described as a form of radiosynthesis, functionally analogous in concept, though not an underlying mechanism, to how plants convert sunlight into energy through photosynthesis. This finding remains an active area of ongoing research rather than a fully settled scientific consensus, but if radiosynthesis functions the way some of this research suggests, it would represent yet another entirely novel energy harvesting strategy that life has apparently developed, this time centered specifically around ionizing radiation as a usable energy source, rather than merely something to be tolerated or survived. Acid mine drainage sites, created when mining activity exposes certain mineral formations to air and water, triggering chemical reactions that produce extremely acidic runoff, sometimes with pH levels low enough to dissolve metal and cause severe chemical burns on contact with human skin, represent another environment long assumed to be effectively sterile due to the sheer chemical hostility involved. The Rio Tinto River in Spain, named for its distinctive reddish color, caused by high iron concentration in its acidic water, has become a particularly well-studied example, after researchers discovered that its extremely acidic water, with pH levels comparable to battery acid in some stretches, supports a surprisingly diverse community of specialized microorganisms that not only tolerate this extreme acidity, but in some cases actively contribute to maintaining it, deriving energy through chemical reactions, involving iron and sulfur compounds present in the water. Research into the Rio Tinto ecosystem has drawn direct interest from astrobiology researchers, partly because the reddish, iron-rich geology and extreme acidity found there bears meaningful resemblance to certain surface features identified on Mars. Through orbital and rover-based observation, making Rio Tinto a useful terrestrial analog site for studying what kind of microbial life might plausibly be able to survive in similarly acidic, iron-rich conditions elsewhere in the solar system, if such conditions turned out to host life at all. Pressure represents yet another axis along which life has repeatedly exceeded expectations about its own limits, and nowhere is this more apparent than in research conducted within the Hadal Zone, the term used for ocean trenches deeper than 6,000 meters, including the Mariana Trench, the deepest known point in any ocean on the planet. Pressure at the bottom of the Mariana Trench exceeds 1,000 atmospheres, a level of crushing force that would instantly destroy most biological structures, particularly cell membranes and the proteins responsible for essential cellular processes, which typically require a fairly specific and stable molecular shape to function correctly, a shape that extreme pressure can distort or collapse entirely. Researchers who have managed to sample water and sediment from these extreme depths using specialized pressure-resistant sampling equipment carried aboard the small handful of submersibles capable of reaching such depths have consistently found active microbial life, and in some cases larger organisms as well, including a translucent, almost ghostly-looking fish species called the Mariana snailfish, discovered living and apparently thriving at depths exceeding 8,000 meters, considerably deeper than researchers had previously assumed vertebrate fish could survive, given the physiological challenges that extreme pressure poses for maintaining stable cell membrane structure and properly functioning proteins. Research into how these deep trench organisms manage to survive via such extreme pressure has identified specialized biochemical adaptations, including unique proteins and cellular membrane compositions specifically evolved to remain stable and functional under conditions that would cause the equivalent structures found in surface-dwelling organisms to fail catastrophically, adding yet another example of life finding a workable biochemical solution to an environmental challenge, long assumed to represent an essentially hard, physically enforced limit on where complex organisms could possibly exist. What makes this accumulating pattern of discovery scientifically significant, rather than simply an interesting collection of biological curiosities, is the direct connection nearly all of it has to astrobiology, the scientific field, concerned with understanding where else in the universe life might plausibly exist and what conditions would be required to support it. For most of the 20th century, the search for extraterrestrial life focused heavily on identifying planets and moons with conditions closely resembling Earth's surface environment, since Earth's surface was, for a long time, essentially the only confirmed example available of an environment capable of supporting life, making it the natural reference point for any search strategy. As extremophile research steadily expanded the known boundaries of what environmental conditions life can tolerate here on Earth, astrobiology researchers correspondingly expanded their own search criteria, increasingly focusing attention on environments elsewhere in the solar system that share meaningful similarities with Earth's most extreme habitable environments, rather than restricting the search only to conditions resembling Earth's comfortable, temperate surface. Jupiter's moon, Europa, and Saturn's moon, Enceladus, both believed to harbor liquid water oceans beneath thick, icy shells, heated not by sunlight but by tidal forces generated through gravitational interaction with their parent planets, have become particularly prominent targets for this reason. Since the chemosynthesis-based ecosystems, events demonstrate a clear, already confirmed biological precedent for life existing in exactly this kind of dark, sunlight-independent, chemically driven environment. Mars, with its cold, dry, radiation-exposed surface and evidence of ancient water activity, has drawn increasing research interest specifically informed by discoveries from terrestrial analog environments like the Atacama Desert and Antarctica's dry valleys, both of which demonstrate that life can persist at extremely low levels of activity in conditions of extreme aridity and radiation exposure closely comparable to what exists on the Martian surface today. This shift in thinking has practical consequences for how space missions are designed and where they focus their search efforts, since a mission designed to look only for conditions resembling comfortable, Earth-like surface habitability would likely overlook exactly the kind of subtle, chemically driven, low-activity habitats, where extremophile research suggests life is actually most likely to be found on a planet or moon lacking Earth's particular combination of sunlight, moderate temperature, and stable liquid water at the surface. Instruments and sampling strategies developed for recent and upcoming Mars rover missions, for instance, have been specifically informed by lessons learned studying endolithic organisms living just beneath rock surfaces in Antarctica's dry valleys, since a rover searching only the exposed outer surface of Martian rocks might miss evidence of a similar, protected microbial community living just beneath that outer layer, in exactly the kind of subtle, easily overlooked niche that surface level. Earth research initially failed to notice as well, until researchers specifically went looking for it after other extremophile discoveries suggested it might be worth checking. There's also a broader philosophical or conceptual shift embedded in this accumulating body of extremophile research, one that goes beyond simply expanding the technical checklist of environmental conditions life can tolerate. Each new discovery in this area chips away a little further at the deeper assumption that life is a fragile, narrowly constrained phenomenon requiring a very specific and limited set of gentle conditions to exist at all, an assumption that made intuitive sense when human observation of biology was largely restricted to the temperate, sunlit, moderate pressure surface environments, where humans themselves have always lived. The accumulated evidence from hydrothermal vents, deep subsurface rock formations, radiation-soaked reactor walls, hyperacidic rivers, hypersaline lakes, bone-dry deserts, crushing ocean trenches, and the near vacuum of space itself increasingly points toward a different, more resilient picture of life, one in which biological processes appear capable of adapting to an extraordinarily wide range of chemical and physical conditions, provided some usable source of energy and at least some minimal, sometimes barely perceptible, amount of accessible water or moisture exists somewhere within that environment, even if only in fleeting, difficult-to-detect amounts. This doesn't mean life is guaranteed to exist wherever these minimal conditions happen to be present, since the actual emergence of life from non-living chemistry remains a separate and still poorly understood question, distinct from questions about survival and tolerance once life already exists. But it does mean that the list of places scientists can confidently rule out as definitely too hostile for any form of life has grown steadily shorter with each passing decade of extremophile research, replaced instead by a growing list of environments that were once assumed to be sterile deserts, geologically speaking, and have since turned out to host thriving, specialized, and often entirely unexpected forms of biological activity. Taken together, these discoveries, spanning boiling hydrothermal vents, kilometers-deep rock formations sealed away from the surface for over 1 billion years, Radiation levels that would be instantly lethal to humans. Deserts. Drier than anywhere else on the planet's surface. Pressures capable of crushing steel and the vacuum of space itself paint a picture of biological resilience that would have seemed like pure speculative fiction to biologists working even just a century ago. Life, it turns out, is considerably better at finding workable solutions to seemingly impossible environmental challenges than the relatively narrow, surface-centered picture of biology that dominated scientific thinking for most of human history ever assumed it could. B. Each of these discoveries individually chips away at a different assumed boundary, and collectively they suggest that the real boundary of where life can exist may still be considerably further out than current research has yet managed to map, a genuinely open scientific frontier rather than a settled question, and one that continues to expand every time researchers develop new tools, capable of looking closely enough at yet another environment previously assumed, with reasonable confidence at the time, to be simply too hostile for anything to survive.